Good day everyone. For today's topic, we are going to discuss about acid-fast staining. Pathologists use various stains to help make their diagnosis. One of the categories of stains is called special stains and refers to a technique where dyes or chemicals are used to identify specific cell structures or components present within the submitted sample. The pathologist can use these particular special stains to confirm or categorize the potential diagnosis. Several special stains can be used to screen for the presence of bacteria. Other special stains can help categorize these bacteria into groups of bacteria. One of the more common special stains used to classify bacteria is acid-fast staining. This stain is used to sort bacteria into acid-fast or non-acid-fast groups. The acid-fast stain determines if the blood, tissue, or other body fluids is infected with mycobacteria, which can cause diseases such as leprosy or tuberculosis, or commonly known as TB, or nocardia, which is a bacteria found in soil and water that may affect the lungs, brain, and skin. According to the World Health Organization, tuberculosis alone is the leading cause of death from an infectious agent, and worldwide, an estimated 9.9 million people became ill with TB in 2020. Tuberculosis is curable. Diagnosis and treatment are critical in the treatment of these diseases. So let's have a brief introduction about AFB staining. So it is a differential staining because it differentiates your acid-fast bacilli from non-acid-fast bacilli. It was first introduced by Paul Ehrlich, later modified by two German doctors, bacteriologist Franz Ziehl in the year 1859 to 1926 and the pathologist Friedrich Nielsen in the year 1854 to 1898. So your AFB staining is used to identify members of the genus Mycobacteria. For the history, it was Robert Koch in the year 1882 who discovered the etiology of tuberculosis. And then later on, Paul Ehrlich developed a stain for TB which he called alum hematoxylin. And then Franz Ziehl altered the mordant by using carbolic acid as the mordant, while Friedrich Nielsen changed the primary stain to carbolfuscin, though using the same mordant. That's why it gave birth to Ziehl and Nielsen's modifications developed by Ziehl-Nielsen stain. Another acid-fast stain was developed by Joseph Kinyon using the Ziehl-Nielsen staining technique but removing the heating step from the procedure. This stain is now the so-called Kinyon stain. Now let's proceed to your acid-fast bacteria. Acid-fast organisms are generally gram-positive, have gram-positive cell wall. So they contain a waxy layer of glycolipids and fatty acids known as your mycolic acid, which is bound to the exterior of the cell wall. So 60% of the cell wall is composed of lipid. So this mycolic acid consists of a strong hydrophobic molecule, making the organism difficult to gram stain. Thus, it makes the organism resistant to acids, giving the name acid-fast. Now let's proceed to the principle of your acid-fast bacilli staining. Acid-fast bacteria, also known as your acid-fast bacilli or simply AFB, are a group of bacteria sharing the characteristic of acid fastness. Acid fastness is a physical property that gives a bacterium the ability to resist decolorization by acids during staining procedures. This means that once the bacterium is stained, it cannot be decolorized using acids, routinely used in the process. So this important and unique feature of certain bacteria gives the ability to classify and detect them using relatively easy laboratory procedures such as microscopy. So in this picture, you can see the difference between the Mycobacterium species and the non-Mycobacterium species as we go under the process of AFB staining. So at first, all bacteria are transparent prior to staining. And then after that, steam is used to force the carbofuscin into all the bacteria. And then the third one, decolorization with acid removes the carbofuscin from the bacteria that do not have the mycolic acid. And lastly, methylene blue is the counter stain. Now let's proceed to the components of your AFB stain. So the primary stain is your carbofuscin, which is a basic dye. So the function of the carbofuscin is that it solubilizes the mycolic acid of the organism. Therefore, if the organism is acid fast, it will become color red. It will absorb the color red. So the time of application for this component is approximately 10 minutes. In order to prepare your carbofuscin, you need to dissolve 50 grams of phenol in 100 ml of 95% ethanol. And then you have to dissolve 3 grams of basic fuchsin in the mixture and add distilled water to bring the volume up to 1 liter. The next component is your mordant. The mordant used depends on the method that is being applied in the sputum smear. So there are two types of methods. The Ziehl-Neelsen method, or also known as the HAT method, uses heat or steam, while the Kenyon method, or also known as the cold method, uses tergitol or phenol as the mordant. So your mordant allows further penetration of carbofuscin into the cytoplasm, or in simpler words, your mordant will serve as a bridge or a link between your primary stain to the cell wall of the microorganism. Next, we have your decolorizer. So this is the component that is being resisted by your acid-fast bacteria. So it is being resisted by your acid-fast bacteria because of the content mycolic acid, which is lipid in nature. And lipids are strongly hydrophobic in their characteristic. So how are we going to prepare your acid alcohol? You need to add 30 ml of hydrochloric acid to 1 liter of 95% denatured alcohol. And then you have to cool it and then mix before using. So the time of application is usually 25 to 30 seconds. Acid-fast cells are not decolorized since they are able to resist the decolorization process of the AFB staining. Non-acid-fast are decolorized. Please take note that the time presented here in your AFB staining will subject to change depending on the protocol of the laboratory or the agency that you are into. And then the last component of our AFB stain is our secondary stain or the counter stain, methylene blue. If methylene blue is not available, you can also use malachite green. So colors of the non-acid fast bacilli are blue or green depending on the stain that is being used. The time of application is 10 seconds. So how are we going to prepare for our methylene blue? You have to dissolve 3 grams of methylene blue in 1 liter of distilled water. So we have the following materials needed in order for us to perform our AFB smear preparation and AFB staining. So you need a clean or sterile glass slide, your sputum sample, pencil or marker, the staining rack, alcohol lamp, AFB staining kit, match or lighter, complete personal protective equipment such as your gloves, your face mask, your lab gowns, your lab shoes, etc. And then applicator stick, and then pasture pipette, and lastly your 2x3 centimeters template. So Mycobacterium tuberculosis is primarily a lung pathogen. Therefore, for all types of tuberculosis, the primary specimen required in acid-fast smear microscopy is the sputum. So there are four chief ways of collecting the sputum, which includes one, coughing, then next is the sputum induction, then the third method is the bronchoscopy, and lastly is via gastric aspiration. So sputum specimens must be collected in appropriate containers. So it can be a 50 ml plastic screw-capped transparent container, which is usually used to foster a secure containment. The transparency of the container allows for visual inspection of the specimen to assess its consistency and quality. Appropriate labeling of the sample with the patient's name and date of the collection must be ensured. The collected sample should be stored at 2 to 8 degrees Celsius until transported to the laboratory. According to the American Centers for Disease Control and Prevention, at least 3 consecutive sputum samples, each collected at 8 to 24-hour intervals with at least one sample being an early morning expectorate, are required for diagnosis. And please take note of your Bartlett's criteria. So your Bartlett's criteria, it is a criteria wherein you are going to accept or reject the sputum specimen, because you do not know that the sample submitted by the patient is not a sputum but instead a saliva. So the Bartlett's criteria is being used. So in order for you to assure that the sample submitted is a sputum, the Bartlett's criteria should comply the following. So under the microscope, you should see greater than 25 polymorphonuclear neutrophils and less than 10 epithelial cells. If that's not the case, then the sample submitted is a saliva. Then you have the right to reject the sample. Now, for the AFB smear preparation, so the recovery of mycobacteria from sputum or mucus-containing specimens contaminated with other organisms is difficult, since mycobacteria generally grow much lower than other bacterial species. The contamination and digestion of the mucus components kills contaminating flora and allows lower growing mycobacteria to grow. Sodium hydroxide in the TB-based digestant, it will act as an emulsifier and a decontaminant, breaking down the mucoid material and inhibiting the growth of contaminants. So we also have NALC, or the N-acetyl L-cysteine. When combined with your sodium hydroxide, it facilitates decontamination by further digesting mucopurulent specimens, which allows the sodium hydroxide to penetrate. We also have sodium citrate. It will aid in the liquefaction by binding heavy metals, thus stabilizing your N-acetyl L-cysteine and allowing it to work properly. So you need to get your new or clean, greaseless, and then scratch slides. And then after that, if there are any identifiers, you should match it on the slide with the clinical specimen. And please take note that labeling should be performed with the material that stays permanently affixed to the slide during your staining procedure. So after processing and concentrating the specimen, 1 to 2 drops of material should be smeared on the slide. And then you need to spread it evenly and smear material in an area of approximately 2 by 3 centimeters. So prior to heat fixing, smears should be allowed to air dry completely within the biological safety cabinet. So if you are not in a biological safety cabinet, you should perform the aseptic technique. So these are the acceptable methods for heat fixing. So the first one is via flame. So you should pass it over the flame 2 to 3 times for a few seconds and then the smear side up. And please take note that you have to avoid the scorching of the sputum that is being smeared. And then the next method is 2 hours minimum at a temperature of 65 to 75 degrees Celsius on an electric slide warmer. And then the next method is 5% phenol in 70% ethanol for 5 minutes. But please take note that this method can kill your acid-fast bacteria. So you have examples here of the sputum smear that is being too thick, and on the right portion we have a smear that is being too thin. So the one in the center is the right amount of the sputum, not too thick and not too thin. Now here is an example of a preparation of a direct smear for AFB microscopy. So it is in courtesy of the QBC channel. So please take note that coiling is not shown here. So I will be teaching you on our on-site meeting on how to coil your AFB smear. So let's take a look at this video.
Preparing a direct smear is a basic step in the laboratory detection of AFB. Prior to smear preparation, choose a new SureFocus slide that is clean and unscratched. Properly label the slide with the patient ID number, as dictated by your laboratory protocol. Once the pulmonary sample has been collected, use a wooden applicator stick to remove a small portion of the mucoid specimen. If the specimen is watery, use a transfer pipette instead, selecting the thicker particles in the specimen, as this is more likely to produce a positive result. However, if the specimen is mostly saliva, request an additional sample, as saliva specimens are not recommended for AFB smears. Transfer the specimen directly to the SureFocus slide, smearing the entire marked circle with the sample. Make sure that the specimen is equally distributed as much as possible. Slides that are too thick or too thin may be difficult to read. Allow the slide to air dry. Once the specimen has dried on the slide, heat fix the slide using an alcohol burner, Bunsen burner, or electrical heating block. If using a burner, pass the slide through the flame 3 to 4 times, being careful not to char the bottom of the slide. Alternatively, when using an electrical heating block, heat fix the slide for 2 hours at 65 to 70 degrees Celsius. Now, the slide is ready to be stained.
Since you already know how to prepare a direct smear for AFB microscopy, now let's proceed to the procedure on how to stain your specimen. So here are the steps. Air dry and heat fix a thin film of microorganisms, and then you have to allow the slide to cool. Next one is to flood the slide with carbolfusin. So steam the slide with the Bunsen burner or alcohol lamp over the sink, then let the slide set for 10 minutes and then rinse with water. Please be careful on rinsing your slide because it may wash off the smear. And then after that one, you have to flood the slide with acid alcohol for 30 seconds and then rinse with water. And then counter stain by flooding the slide with methylene blue for 10 seconds. Then rinse with water. And then next is to dry the slide by blotting on a piece of paper. And lastly is to view the organisms using the oil immersion objective of your microscope. Again, please take note that these procedures are subject to change depending on the protocols of a laboratory that you are assigned into. So here is a quick video of using the Ziehl-Neelsen method on how to stain your AFB smears. So let's take a look. Alright, that video was in courtesy of Jimmers College, so from YouTube. So now you already know how to perform the AFB staining, so you are ready to do the activity on our meeting. So let's continue. So here are the different AFB staining reactions. So we have acid-fast bacilli, which is color red, and then non-acid-fast organisms are color blue. Acid-fast bacilli are color red due to the absorbance of your carbolfucin, which penetrates the cell wall because during the decolorization process, the acid alcohol was resisted. That's why it retains the primary stain. Unlike with your non-acid fast organisms, they don't have the mycolic acid in their cell wall. Carbolfucin was being washed off during your decolorization process or upon the addition of your acid alcohol. That is why your methylene blue now penetrates the cell wall of your non-acid fast organisms. That's why they are color blue. So the background of your AFB stain under the microscope, it depends on the secondary stain that is being used. But please take note, AFB are color red or red bacilli in a blue background, while non-AFB organisms are color blue on a blue background. So bacteria displaying acid fastness includes the genus Mycobacterium and then the genus Nocardia. So under the genus Mycobacterium, you have Mycobacterium leprae, Mycobacterium tuberculosis, Mycobacterium smegmatis, Mycobacterium avium complex, and your Mycobacterium kansasi. For the genus Nocardia, you have Nocardia brasiliensis, you have Nocardia syriacigeorgica, you have your Nocardia farcinica, and your Nocardia nova. But please take note, acid fastness can also be attributed to other structures not classified as bacteria. So this includes your bacterial endospores, the head of the sperm, the Cryptosporidium parvum, Isospora belli, your Cyclospora cayetanensis, Taenia saginata eggs, your hydatid cyst, Sarcocystis, nuclear inclusion bodies which is found in lead poisoning. Even though acid fastness can be attributed to many different bacteria, correlation with history makes it a fairly unique characteristic of your Mycobacterium tuberculosis in clinical practice. So this makes acid fast staining sensitive and specific provided clinical correlation is part of the equation. Now let's proceed to the treatment. So for the first line of drugs, so we have the mnemonics RIPES. For the second line of drugs for TB, we have the mnemonic CC Coke. So yes, TB can be cured if the full course of the prescribed drugs is taken regularly and without interruption. So the World Health Organization approved standardized and effective cure for TB called DOTS or the so-called TB DOTS. DOT stands for Directly Observed Treatment Short Course. It is available in all countries of the Southeast Asian region. It takes at least 6 to 8 months of medication to completely treat that disease. So it is very important that the patient takes all the prescribed drugs for the recommended duration. So it is dangerous both for the patient, family members, and the community if he or she stops taking medication before the prescribed course is completed and without medical advice. So the TB germs that are still alive become even stronger or resistant to the drugs. So stronger drugs are then needed to kill these super or TB germs if the treatment is not completed the first time. So we also have the terms MDR-TB and XDR-TB. So MDR-TB and XDR-TB is done via the use of your polymerase chain reaction or the GeneXpert. Okay. So the screening test for your TB, you have your DSSM or the direct sputum smear microscopy, and then we also have the tuberculin test, or the Mantoux test. And then for the confirmatory, we have the for culture, you can use your culture such as your Lowenstein-Jensen. And then for the polymerase chain reaction or the PCR is this one, the Gene Expert, which identifies or classifies your TB into MDR-TB or XDR-TB. MDR-TB stands for multi-drug resistant tuberculosis, and then XDR-TB stands for extremely drug resistant tuberculosis. So MTR-TB caused by an organism that is resistant to at least isoniazid and rifampin or rifampicin. And then XDR-TB or the extremely drug-resistant TB is caused by an organism that is resistant to isoniazid, rifampin or rifampicin, and the fluoroquinolone or by an organism that is resistant to isoniazid, rifampin or rifampicin, and a second-line injectable such as your amikacin, capreomycin, and kanamycin. So that ends my presentation for today, everyone. So hopefully that you have learned something new for acid-fast bacilli staining. For questions and clarifications, please don't hesitate to message me or ask me during our on-site schedule. Thank you so much and have a good day. God bless and Bismillah.